My name is Wilmer Cuellar, and I will present the progress on Objective 3. The activities in this objective are possible thanks to the close collaboration we have with these different teams in the region and the support we have received from the team in Colombia, validating different protocols that we are implementing in Asia. We have focused on these two diseases, Cassava mosaic disease and Cassava witch's broom disease. And as you can see, the symptoms associated with these diseases are distinct, and they can be easily identified from the photographic record, as you are doing now. To organize the information coming from the different groups, we have built a project page within the Best Displays platform where we can store the images, we can revisit them, we can check for different kind of symptoms, and we can update this information with the results coming from the laboratory. So far, we have collected almost 40,000 images in this using a standard protocol. In 2020, we managed to collect 28,920 images, but due to COVID restrictions, travel restrictions in 2021, we were able to collect almost only 10,970 observations. The data is available to all the participants, which also collaborate in the analysis and curation of the data. As I mentioned before, we have focused only on CMD and Cassava witch's broom disease. The map you are seeing now is for CMD, where the yellow color indicates the presence of the symptoms of CMD. When we analyze this data at the province level, the incidences show that CMD is dominant in Cambodia and the south of Vietnam. There are some pockets now in the south of Laos and in the north of Vietnam, but the situation is completely different to that observed in 2014 when the last region-wide survey was carried on. Almost the opposite seems to be the case for Cassava witch's broom disease, which in 2014 was very dominant. It has incidences of more than 50% in some provinces. But in 2020, we don't see these high incidences. We see pockets of the disease in Cambodia and the south of Vietnam and in Laos, but nothing close to the 50% incidences in any of the provinces that have been surveyed. The reasons behind this are under analysis now, but this seems to be the case as we review the survey data done at country level at different times by our teams in the region. I'm quoting here the work from Minato, for example, in 2017 in Cambodia and Vietnam, and Siriban in Thailand in 2019. Although the symptoms of Cassava witch's broom are very easy to identify, this is not the case with the detection of the phytoplasma associated with this disease. As you can see here in red, we show all the sequences that belong to phytoplasmas found in Cassava witch's broom disease plants. And these sequences belong to at least 3 different phylogenetic groups according to the current classification methods of phytoplasmas. So if this diversity has implications in the efficiency of the detection methods available for phytoplasma associated with Cassava witch's broom is not known yet. We have evaluated some of the nested PCR tests available, and the results are not promising. As you can see here, the number in red shows all the samples that show symptoms of the witch's broom disease, but are negative to the PCR tests. And although there are more than 20% of plants that show positive PCR, but no symptoms of witch's broom disease, Most of these bands do not correspond to phytoplasma sequences. They are of different bacteria, and there is no clear trend there. So there is still work to do in the detection of phytoplasma and cassava disease. The opposite is the case with the detection of the virus associated with cassava mosaic disease in Southeast Asia. We have standardized CTAB and dipstick protocols. The team in Australia has been working on a droid to detect Sri Lanka cassava mosaic virus in the field. And also we have been working on the nanopore protocol to detect and sequence the complete genome of the virus. And because we are using random primers here to amplify the circular DNA, we should be able to detect if there is any other Gemini virus present in the region. So far, we found only Sri Lanka cassava mosaic virus, but at least two subgroups can be observed already. These are indicated here in the red circles. The difference among these two groups is the presence of a red gene longer than expected for some of the isolates. If this has implication in the virulence of the pathogen is not known. There are some reports that indicate that this region has a role to play there, but we have not tested this in our. We apply sequencing also to the identification of the whiteflies that we can collect in the fields of Cambodia, Vietnam, and Laos. And so far, for two of these countries, we found Asia 2.1 and Asia 2.6 as the major biotypes in cassava, colonizing cassava. We know that Asia 2.1 is quite an efficient vector of Sri Lanka cassava mosaic virus compared to other Bemisias found in the region. But the role of Asia 2.6 is not known yet. And if there is any association between these biotypes and the abundance of whiteflies or the incidences of CMD is something that is under analysis, and Maria Isabel will be showing some of the results from those analyses during this review. I think all this information has been very efficiently used by the teams in the region to raise awareness about the problem at the regional level. And because they have access to all this information, and they could use it to mobilize resources and to plan rapid responses to the spread of this disease. One of our roles during this project has been also in support the capacity building of these researchers in Southeast Asia. And we have done this during these years, not only by training in the different protocols that we have described in this presentation, but also in mentoring their participation in international conferences and in supporting their participation, active participation in the preparation of scientific publications. I think all of these will reflect very positively in their future work as well. In summary, I think diagnostic protocols for CMD have been validated, and the next step would be to implement those during the field surveys in 2022. We are also thinking of implementing the nanopore sequencing in the region. The surveillance data has mapped regions of high and low disease incidence, and this could be useful information for the teams working in the other objectives of the project. And the apparent decrease in the incidence of cassava witch's broom disease needs to be validated. We are considering to include genotyping data to see what if there is some difference between these pockets of high incidence of witch's broom disease and regions where we find no disease. It's clear that we need to improve the molecular identification of the phytoplasma, and for this we think that the genome sequencing of different isolates would help us to identify better targets for detection. The two biotypes that have been identified in Vietnam and Laos should be further confirmed for Cambodia. The samples are available, but they need to be analyzed. The contribution of the different biotypes to the transmission and dissemination of CMD is under study and should be considered for the next half of the project. Thank you very much.